Thank you for patience, and also a great honor for me to be here, particularly on site in earlier of this morning, to witness the great success of the BGI's new invented technology, the Stereo-seq. So I believe that Stereo-seq is opening the new angles to understand many biological questions in different way. So I'm Yuhui, working at the Southern University of Science Technology, and also a core PI at the Shenzhen Key Laboratory of Gene Regulation and Systems Biology. So gene regulation actually is a fundamental to almost all the living organisms. So in our key lab, so together with other groups in the university, we are interested in systematically deciphering the gene regulation networks at different topics, from differentiation to development to human disease and so forth. So global regulation investigation on gene regulation requires a larger collection of expression data, which can only be achieved through multidisciplinary interactions involving technology development and also large efforts on the computational development. So in the past around 2 decades after the completion of the Human Genome Project, we have altogether witnessed the great inventions on technology for development. For example, the microarray chip that can be used for large scale gene expression profiling, and also the second generation and also the third generation of DNA sequencers. And also here, right here, BGI is also one of the world leading DNA sequencer manufacturers. Also, these technology inventions in turn pushed the fast advance of the new software development. And altogether, many new biological insights has been disclosed. After that, as many talks you have heard in this morning, the single cell technology is without doubt one of the, I think, one of the most important new technology inventions in less than 10 years. So with the continuous emergence of new methods, we can see that the scale of single cell experiment constantly goes up, from at the beginning, the manual picking of the single cells to multi-well plates, and also later higher throughput with the nano drops on the picowells. But from biological point of view, single cell sequencing also has been widely applied to almost all the level of molecular information flow. For example, from DNA to RNA to protein, also including chromosome structures and some epigenetic markers. In this slide, in this view, list some, but not all of the single cell sequencing methods, all of which has specific advantage and also disadvantages. So in our lab, recently, we developed a single cell compatible method called the CASP for single cell sample multiplexing. CASP utilizes the general membrane binding characteristic of a protein called the concatenavalin A, which incorporates with the oligobarcode tailings that are compatible with single cell RNA-seq and also single cell ATAC-seq. So CASP with high throughput sample multiplexing, CASP can largely reduce the single cell costs. So very recently, so following the similar approach, we use a cell damaging molecule probe and develop a new method called DISC-seq. So this method is designed for quantitative measurement of the cell death or cell damage signal rates from at a single level incompatible or together with a single cell RNA-seq. So of course, today I'm not going to talk about in detail of these technologies. Today is a day of the Stereo-seq. So beyond the great success of single-cell, routine single-cell technology I just mentioned, one very important information layer is missing. That is the actual positional information of one cell, and hopefully even one single transcript within one cell. So as named as a method of the year 2020, the scientists are keen to obtain captured spatially resolved transcriptome data. So this cover story just came out exactly at around several months after we initiated the spatial transcriptome project on fly. So firstly, why fly? So Drosophila has a special and relatively short life cycles. It has long been used as a successful model organism to study many biological developmental questions. And second, the tissue-specific marker genes of fly are readily available, and also the database containing tissue-specific transcriptome data also online, including 2 versions of Fly Atlas, DGET. But all these expression patterns are limited at the tissue level and lack the concrete cellular positional information. So from the 3D view of the expression data, there are much less resources available. The virtual fly brain is a database integrating the expression and the virtual 3D expression data that is from the published literatures, and also a recent paper from Schaefer and their colleagues. They promoted or generated the first complete map of the fly central brain neurons and their connections, and by scanning the electron microscope images. And also on fly embryo, there was just, actually, they are the former colleague, my former colleague at Berlin. They generated the first 3D expression map of the first of one stage fly embryo by combining or integrating single cell RNA-seq together with in situ hybridization data. And the 3 expression level are at singular cell level, but as they claimed as a virtual 3D map. So the exact position of the transcript are just computationally predicted. And also in our work, they performed only one stage fly embryo. So the temporal information of the entire embryogenesis process are largely missing. So above all this reason I just mentioned, one more important reason also needs to mention. The postdoc, Chinan, my postdoc, he's fascinated by these small animals flying out of the smelly fruits, particularly in their egg activation and early development. So both procedures are highly characterized with a positional, localized gene expression and regulation. So we actually start with manually cutting the tiny small pieces into cryo sections and trying to sequence the slice transcriptome. But the process was slow and difficult. So it was lucky that we have a long collaboration history and with BGI people. And also it was exactly the time when they invented the Stereo-seq. So then Chinan starts to working with this group of people from BGI. They are young generation, enthusiastic, and work very hard and always with smiley faces. So goes to the detail of the project. I think my introduction today on Stereo-seq is definitely unnecessary. Here, I just want to emphasize that for a barely seenable embryo, fly embryo slice, there has been over 200,000 DNB beans that can be covered. So in our piloting experiment, we have performed Stereo-seq on the last 2 stages of embryo and as well as 3 stages of larva. So as when we merged 20 times 20 beans together, that equals to almost the size of 1 single cell. Over 1,000 transcripts could be captured with high quality, corresponding to a few hundred of genes. So the gene number is relatively small compared to single-cell RNA-seq, standard single-cell RNA-seq, but nevertheless, the cells can be nicely clustered together and displaying many tissue-specific marker genes, as you can see here by the heat map. And this slide shows comparative results of Stereo-seq with in situ hybridization data. As you can clearly see that in the first and third column, the results from the Stereo-seq are highly consistent with the second and fourth column, the results from the in situ hybridization data. So this means that Stereo-seq can faithfully and efficiently capture the localized expression patterns. And moreover, in our project, so Stereo-seq also identifies new tissue marker genes with relatively low expression level. So sometimes small is good. So in our project, we could consecutively sequence each sections of the embryo samples so that with this data, we could rebuild the 3D expression model. At the beginning, we used the existing softwares like 3D slicer and paste. So at the end, we could get the 3D expression model. As this slide shows, you could say that the contour and the structure of each tissues resemble very nicely with anatomic position or anatomical true positions, as here the central nervous system, the fat body, or the midgut. And with these models, we could do many further, I think, exploration, such as we could identify the subregion of organ. For example, in the left panel is the results from the midgut. We could identify with different color here, the different subregions. And also we could track the cell state dynamics and also the expression trajectories along with the developmental stage. And also by applying cynic analysis, we could also gain the localized expression regulation of the transcription factor regular activities. So due to the time limitation, I cannot go deeper into these details. Then also our results has been published just last evening. So encourage people to just read this paper. I just want to emphasize that from publication of paper is not the end of the story, but rather a start of even long journey. So from our early experiment, some technical and analytical challenges were realized. For example, these similarities are sometimes observed between 3D reconstructed models and actual anatomic structures. We assume that this is mainly due to the uncertainty in defining the single cell boundaries. Therefore, in our new experiment, we developed the nuclear eye staining protocol and took the image captures before Stereo-seq. And also Stereo-seq captures relatively low RNA molecules in total. Therefore, some rare or sparse types are more difficult to be identified. So, and currently we are also performed routine single cell RNA-seq on each biological samples in parallel. And also our previous publication contains only the results from last 2 embryo stage and larva. And we are now expanding the experiment to the entire developmental stages, as well as larva and the pupa. So just quickly go through the improved results from our newest data. In these slides, you can see that, the up panels, old results, and the lower, obviously, you can see that through improved protocol, this cell nuclear staining protocol, and the cells could be much better identified. So with the clearer cell boundary, we could actually cluster cells in much better resolution. And as a result, the tissue reconstruction also offers a higher resolution. And this slide here shows at just 2D level to looking at the cluster of tissues. And also, this new data allows us to improve the 3D remodeling reconstruction results. As you can see here in these slides, that the results from central nervous system, somatic muscle, and intestinal tracts, the reconstruction results actually offers a much higher resolution. So we merging all of these tissues together, we could actually view or reconstruct the whole body with the actual position of each tissue, each cell, and also individual genes. So in the last slides, here just show a few examples of the genes and their localized expression at the 2D level in the left panel, and also their localized expression at 3D level and in the right panel. So to summarize, that Stereo-seq provides unprecedented resolution and throughput for spatial transcriptome profiling. So in our project on fly, beyond the normal 2D analysis pipeline, we also built up a 3D analysis pipeline to rebuild the entire spatial temporal developmental these stages. And also, we are expanding the experiment on the entire embryo stages with less than 1 hour time intervals, and as well as 3 stages of larva and pupa. So together with the standard single-cell RNA, by the end of the project, we expect to see the entire spatial temporal dynamics of the entire developing cycles of life, except the adults. In parallel, so the new analysis tools that are powered with new deep learning method are also under development together with collaborators. And also we believe that with such vast resource of data, many biological questions will be addressed or answered, and of course supported with functional validations. And we also are interested in review some evolutionary conserved gene recognition networks by comparing to other bigger animals like mouse. And also we are actually now building the disease models on fly, trying to understand the altered spatial information or patterns under the disease development. So generally speaking, my lab actually focus are not limited to fly. So cancer is also the research we are working on. So we are currently also applying SpatioTemporal single-cell technologies in cancer clinical patients. So here just take this opportunity to put an ad. So talented and diligent scientists are welcome to join. So and then just well, stop here by acknowledging people that are contributing to projects I just mentioned today. So Qinan, as I mentioned, he's a leading scientist dealing with the wet experiment of the fly and also single-cell RNA-seq of disc-seq. So Wei Chen and a few other members from his lab is contributing to establishing the CASP and the disc-seq methods. And finally, a very special acknowledgement goes to the BGI, bunch of BGI peoples. With their hard work in the last 1.5 years, they made this project successful. And currently, we are working together, continue to working together and hardly to make this project move forward and hopefully bigger. So finally, thank you for your listening.
Thanks.